And hello, everyone, and thank you so much for having me. I will be talking about the ProteinFold, the Australian ProteinFold service. And I would like to highlight that I'm presenting this project on behalf of a large group. But before I dive into the details, I would like to introduce Australian BioCommons. So we support Australian life science researchers by providing them to access to community-scale digital infrastructure, including software and hardware platforms. We operate several services. They are used for different purposes. We are nationally funded, or our services are fully subsidized for Australian researchers. And we also have training and support solutions. And one of our main services is the Australian Nextflow Seqera Service, which is we have our own platform, Seqera platform, and we host and onboard the Australian researchers into the platform itself and give them access to different workspaces. Also, they can have their own organizations. And what we are working on as well is to have like some commonly used workflows, deploy them on the platform, but also provide the compute infrastructure and behind. So from the user perspective, the only thing they need to do is to bring their own data, log into the service, run the workflows on our compute infrastructure, take the results, and use them in any way they want. And the way we usually work is we engage with different life science communities that can maybe focus on specific methods like genomics or single-cell omics, or consortia that focus on a specific species like the Childhood Cancer Institute, but also Genomics for Australian Plants, and as well as data production facility. And the aim of this engagement is to identify common problems they have. Then from our side, we try to find some solutions that avoid these problems if possible, but at least mitigate the impact of these problems. And in a latest engagement with the social biology computing community in Australia, we realized that they have a lot of challenges with relating to using the tools in their fields, and mainly because these tools are compute intense. They need a lot of GPUs, and this is not accessible for everyone. And in many situations, it's also a bit a bit difficult to understand, especially for non-technical users. The whole field is developing very quickly. That makes it very difficult for the researchers to follow up with all the advances. And there is like wide significant applications in this field. And therefore our response was to focus on different aspects, including improving the communication within the community, facilitate the exchange of information and knowledge, establishing platforms to make it easier for them to use any tool they want, and also supporting the infrastructure side and providing documentations and user guides. And in this talk, I will focus more on the platforms side. One of the main problems they work on is the ProteinFold problem, and simply it's just predicting the protein structure from the amino acid sequence. There has been recently many tools that were published, and they were published in high-impact journals like AlphaFold, the ESMFold, ColabFold. And what we wanted to do is to facilitate for the users the access to these tools. And this problem is very significant. The authors of AlphaFold won the Nobel Prize for this contribution. And that's what took us more to thinking on how we have a platform. And considering that we had the Seqera platform, so we wanted to have the platform based on Seqera, and make it easier for the users to run the tools, considering that the users can come from different backgrounds, different technical expertise, make it easy for them to access these tools, run them from the platform, and also on different compute infrastructures. Either what we provide, but also if they have their own infrastructure, they can just utilize them for this purpose. And this is what led us to go more to the ProteinFold NF-core workflow. And we thought that's our starting point because, first of all, this workflow is maintained by the NF-core team and the Notre Dame lab people. So we know it's written in best practice, easy to extend, and it has almost all the dependencies we need. So it's easy to deploy on different infrastructure we have. And also it supports the main models we want at this stage, which was the AlphaFold 2, ESMFold, ColabFold. So we thought that's a good starting point for us. And that's what led us actually to reach out to the maintainers here in the CRG, and mainly Jose and Cedric. And we said, well, we think we have the same goals. Maybe we work together, unify our efforts, and contribute to this workflow And all our ideas were welcomed, and we started to work together on this workflow. And some of the improvement we done recently is to allow running the multiple models in parallel on the same samples. Then we added multiple visualization components just to make it easier for the user from the platform to interact with the results and do some benchmarking. And we also added another feature that allows the users to search for the predicted structures from these models and compare them with known structures in some databases and find the most similar structures. And I will show you in a demo. We recorded some of these details. Yeah. So this is our own instance of the service. But what applies to our instance will apply to any Seqera platform we have access to. And I will just highlight that when the user is at the beginning want to launch this workflow on the platform, they will get this user interface that allows them to customize all the parameters of the workflow, including the general ones like the input, output, the sample sheets, and all this stuff. But also you could go to more details and customize the parameters of each service. So it allows you to have a comprehensive customized execution of the workflow on the infrastructure you want. I'm not going to do any executions here. I will just show you what we get from a completed execution. And so this execution was done using the development version of the ProteinFold on NFCore. This is the main page where when the workflow is running, you will see all the information about the whole workflow and the processes. This is a successful workflow, even when the process has failed, but in the retry, it passed without problems. And you could get all the information, including the aggregated statistics about the run, but I will focus more as well on the report you will get. So after a successful run, you will get access to different reports. One of them is the visualization of the predicted structures by each model. And when running multiple models in parallel, you will get one report per model per sample. And this is an example of what you will see when running, for example, AlphaFold. Just directly from the platforms, AlphaFold will produce 5 different models. The users will have a visualization of these models in the 3-dimensional structure. They can interact with them. They can see some general information about the samples, can zoom in, zoom out, rotate. The models even navigate between different ones. You can see the PLDDT confidence as well reflected on the structure. You could even change the representation of the models. I think 4 of them are supported. And also we report all the confidence metrics that people are interested to see, including the PLDDT plot. That's the predicted local distance difference test that shows you the confidence in the local structure and per amino site. But also you see the sequence coverage, so the number of sequences that aligns to the input sequence as well. And we also have links from the report itself to the GitHub web pages of the community that has a lot of resources, external resources, or some resources developed by the community that gives you more information on the models, the problem itself, how to use these models, what do you need to look after in the results, just to make it easier for the users, if they're seeking more information, that they can just quickly jump to these web pages directly from the results and take a look. The other report we generate is when running multiple models, we take this, the predicted structure from the different models and align them together, allowing the users to have a visual benchmarking of running the different models on the same sample. And in this case specifically, we run the AlphaFold, ESMFold, ColabFold on the same sample. You see how all the 3 structures are aligning together. The users can, pick and choose any combinations of the models to compare between them. The time you have only one model selected, you go back to the confidence coloring, so you understand more about the confidence in the local scale of the workflow. Again, you go down and you see the similar plots. You get the PLDDT, the sequence coverage. ESMFold does not produce this plot as they don't do this step, but you will see it for AlphaFold and ColabFold. You could navigate between the different samples, downloading the PDBs directly from the platform. And the third report you will get is the fold-seq output. And simply, we just have a report where we show, in this case, because we compared with the data with the PDB databases, it shows that there is like around 94 hits in the database. It shows you the similarity in the sequences from any of the hits you got through fold-seq easy search. It also shows you the alignment of the predicted structure with the one that we are like checking at this time to see how they align together. And although you see all this stuff on the platform, you could just run the workflow independently on the command line, and all these reports will be generated, and you can just access them directly on your local PC. In this case, I will just download the fold-seq report and open it in my browser. But if you do it simply with the command line, you'll still be accessing all these reports, and you can share them as well. This is an ongoing project, and the way we want to work is more to listen to the users and the researchers and try to accommodate their needs and prioritize them. We have some ideas to include RoseTTAFold and RF diffusion in the future. We will be a lot of work on the improvement of all this visualization report and benchmarking. And this is an open source project. Anyone, especially the workflow itself, anyone is welcome to contribute. We appreciate it. If you have any ideas and you want to contribute, you're very welcome. Again, this is international collaboration. We have a lot of organizations involved in this project in Australia and different states, but here as well in the CRG. And more specifically, these are the people who were involved in this work. And I will thank specifically Jose for maintaining the workflow and facilitate all the collaboration we had internationally, considering the different time zone. Cedric as well for supporting our collaboration. Kate Mitchie is our champion of the community that helped us, to understand more about the user's needs, and Min implemented all these visuals you saw in HTML. Thank you so much.